Okay, so today's topic is we're now out of the classification naming section of this unit. So the next topic now is viruses, which is very relevant for today's topics. So I'm going to start presenting. Actually, hang on, just give me a second. I just got to get it downloaded and set, and then I can present it. Okay. So present now, tab. There it is, viruses. Okay, so viruses. So these are the topics we're going to cover. So prokaryote versus eukaryote. What are viruses? Virus replication and treatment. I just want to see if it kind of gives you background. Okay, yeah, it does. Okay, so I just wanted to go ahead. So prokaryote versus eukaryote. So these are two basic types of cells. Prokaryote, you got the pro, which means before, and then karyon means nucleus. So before nucleus. So prokaryote lacks a true nucleus and organelles. Okay, so an example of these is the bacteria and archaea. So remember, those are two of the kingdoms when we were talking about the 6 kingdoms going back. So your 6 kingdoms being your Animalia, Plantae, Fungi, Protista. And then now we're talking about archaea bacteria and eubacteria. Not totally talking about it today because we do our bacteria on Friday, but those are two of those kingdoms. Bacteria, are eubacteria, archaea bacteria. Those are two of the kingdoms. Okay, and those are examples where we lack the nucleus. Okay, so I went longer than I need to go there. So that's for prokaryote. Now eukaryote, so the U means true, karyon means nucleus. So now we have true nucleus. So it contains a nucleus and membrane bound organelles, right? The organelles being your mitochondria, ribosomes, and so on. Example of this is your plant and animal cells. Okay, plant and animal cells have the nucleus. Your prokaryote lack nucleus, and that's what your bacteria and archaea bacteria are. Okay. Okay. So now going forward, looking at prokaryote versus eukaryote. So prokaryote, the one that lacks the nucleus, small, which makes sense because they don't have the nucleus that takes up all that space. Your eukaryote are large because they have the nucleus. Now, prokaryote still has DNA, but it's not bounded by a membrane, whereas the eukaryote has the DNA, which is found in your nucleus. You should remember that when we did mitosis, all the DNA is found in the nucleus of the cell. Remember, it's that chromatin. That's your DNA. So prokaryote just has single chromosome. Asexual reproduction, right? So you don't need that whole sexual reproduction where you have to have the, in this case, like the egg and the sperm come together to create, right? So you don't have to have that when it's asexual. It just reproduces on its own. Whereas eukaryote, you have to have that sexual reproduction, which is your mitosis, meiosis. So eukaryote, most are multicellular, right? We're talking about animal cells, plant cells. We have more than one cell making us up. Whereas eukaryote, that bacteria, not necessarily are they multicellular. Okay, I'm just going to scroll down a second here. And then no membrane bound organelles, right? So you have nothing that's bound tightly. And then with your eukaryote, they are bound, membrane bound, because they're within, the cellular membrane, which keeps them all together there. Okay. So, Yeah, I don't want to get too. So eubacteria, um, which is that eukaryote, or which is the prokaryote is the eubacteria and archaeobacteria. Okay. Um, they both can thrive in harsh habitats. Um, the archaeobacteria does have a cell wall and ribosomes again that make up very different eubacteria. Um, and they have different substances inside of them that we will get to when we get talking about the bacteria on its own. Um, many are anaerobic, the prokaryote, and then the eukaryote are aerobic. All right, so talking about these, leading us into talking about viruses, okay? First thing we're going to look at. So we talked about prokaryote and eukaryote cells, right? Having those nucleuses, and cells make up living things, right? The prokaryote make up our bacteria, which I'm going to go back to. The eubacteria and archaeobacteria, that is one of the kingdoms, right? Those kingdoms, the classification of living things that the kingdoms are, right? The kingdoms were the levels of, was that top level of classification. That was number one. Those were the broadest ones, right? So lots of different living organisms were under each of those kingdoms, and there was 6 of them. All right. Bacteria actually was 2 of them. You had the eubacteria and the archaeobacteria. So there's actually 2 different kingdoms that have that are bacteria related. But was there a kingdom that had viruses in them? Was virus part of any of those kingdoms? You know, you had your protista, you had your animalia, you had your plantae. Was virus among them? So is a virus considered living or dead? Okay. So going down here, viruses do not appear in the 6th kingdom classification because they are considered to be between living and non-living. Almost like a zombie, I guess. Just kidding. Obviously not a zombie. Anyways, okay. So it's kind of in between the living and non-living. And we're going to get into specific reasons why. Okay. So a virus is a lifeless particle that carries out no metabolic functions on its own. Okay. So just going on, it cannot reproduce on its own unless inside a host cell. Okay. And we'll get more talking about this. But really, what a virus does is it attaches itself to a cell. Okay. And that's a living, human cell. Or I don't want to say human, animalia cell. I guess they've said now that they've found animals that now have coronavirus. So I guess it doesn't necessarily need to be a human cell. So I'm just going to scroll down here. So what is a virus? Okay, so viruses were not seen until 1950 when scientists developed an early electron microscope. And this enabled scientists to see viruses. Okay, so being a tennis ball being this big, cancer cell, bacterium, virus is smaller than bacterium. So that's 10 to the power 2. And this is nanometers here. Okay. So again, a virus is so small that it must be measured in units called nanometers. And 1 nanometer actually equals 1 billionth of a meter. So that's 10 to the negative 9. Okay. Which means that if you were to take 10 with the decimal, you would move the decimal 9 places to the left. And that's how big a nanometer is of a meter. So that's like, 9 zeros, 8 zeros in front of the 1, 8 zeros in front of the 1. Okay. And so viruses range in sizes from about 20 nanometers to 400 nanometers in diameter, which again is still super small. So you cannot see them unless you have a very strong microscope. Okay. So because it is so actually just as a visualization, so difficult to visualize such small measurements, imagine more than 5,000 influenza viruses fitting on the head of a pin. Okay, that's how small they are. So when you think about that, 5,000 influenza viruses on the end of a pin, they're so tiny that you have no idea where they are, right? So you have no idea where to pick them up. Okay, let's stop talking about how small it is and let's keep moving forward here. Okay. So what is a virus? So viruses are actually far less complex than the simplest living organism. It consists of it's two parts that you can see here, right? It consists of like an inner nucleic acid, which is your DNA or RNA core. Okay. That's your nucleic acid, which we talked about in the very first unit. And it's like a core or a strand. And then it's surrounded by an outer protective protein coat. Okay. Which is called the capsid. So that's here. This is your capsid. And this is your protein coat. So you have your DNA up here, your RNA. And then around it is this capsid protein coat. And this is what gives the virus its shape. And they don't all have the same shape type thing. But so you have this protein coat, that's the capsid. But the capsid actually accounts for 95% of the total virus. And it gives, obviously, and the focus of it is just to give the virus its shape. Okay, so bacteriophages, also referred to as phages, are a category of viruses known as eaters of bacteria. Okay, so obviously the name bacteriophages, they're eaters of the bacteria. They have a unique tadpole shape with a distinct head and tail. And you can kind of see that this one has this here. This is kind of, I think, representing the bacteriophage. This is what a bacteriophage would look, where it has the head and the tail. And this is a virus that actually attacks bacteria. And when we talk about bacteria, you'll actually learn that there is good bacteria and so helpful, actually not good and bad. I'll say helpful bacteria and harmful bacteria. Okay, so there is actually two different types of bacteria here. Okay, so let's keep going here. Oh, so there we go, talking about the bacteriophages. Okay, so this is bacteriophage here. This is your virus. So there's the DNA inside the head, and then you have this protein capsid over top of it. And these are the bacteria eaters. So I already talked about that. Sorry, I forgot that was in here. Are all viruses deadly? No. An example is tobacco mosaic virus. So it infects the leaves of a tobacco. Why do I find that word hard to say? A tobacco plant, but does not appear to destroy the plant tissues. That's just an example. I'm sure if we looked into it, there's a lot more viruses that are not harmful or killers. So viruses are selective and only infect specific cells. Okay. This is where we start talking about a host. So a host range, the limited number of host species, tissues, or cells that a virus or other parasites can affect. Okay. Some animal viruses have a broad host range and can go to lots of different hosts. For example, the swine flu virus can infect hogs and humans. So swine flu didn't just affect humans; it also affected hogs. The rabies virus can infect many mammals, including rodents, dogs, and humans. And then other viruses have a very narrow host range. For example, the human cold virus actually usually affects only the cells of the upper respiratory tract. Okay, that human cold, that's, you feel it in your chest. That's only affecting the upper respiratory tract. Whereas the HIV okay, and that is a virus, the V stands for virus, affects the immune system because it attaches only to a specific site on the surface of certain types of white blood cells. Okay, so the HIV virus only affects the white blood cells. Okay. All right, so just going back here. Viruses, I should really look forward to this because I just talked about this. So wide host range, so rabies affects mammals, dogs, rodents, humans, just mentioned that. Narrow host range, human cold affects cells of upper respiratory tract. HIV affects the immune system. Okay. Okay. So now looking at viral replication. Okay. So this is how the virus spreads. Okay. So there's 4 basic steps of replication here. Okay. So the replication is actually the process by which the virus and its genetic material, right? Because there is that DNA in the head of the virus. So genetic material, how they duplicate before a cell divides. So this occurs in a variety of ways, but there's 4 steps, which we see here. There's 4 basic steps. Okay, going into the picture after. Okay, so first off, the virus chemically recognizes a host cell, attaches, and invades. So really what's happening here, it's attachment and entrance. Okay, that's step 1. So the virus chemically recognizes a host cell, attaches to it, and then enters it. Okay, so either the whole virus or only its genetic material, that's for certain viruses, enters the cell's cytoplasm. So you know that the cell cytoplasm is what? It's like the filler of the cell, right? That what fills the cell up. And so again, talking about the host cell, think about when coronavirus first happened. They found that it wasn't affecting kids, right? It was mostly affecting adults. Okay. So obviously the very first coronavirus, because obviously we've had a few variations, the host cell was more towards adult cells. Okay. So the second step, DNA or RNA directs host cell in replicating viral components. So the synthesis of protein and nucleic acids, molecular information in the viral DNA or RNA. So this is within that head of the virus, the head where the DNA or RNA are. It directs the host cell in replicating the viral components. Okay. So that's just, it's kind of like it takes over the cell and it forces the cell to start replicating the virus components. Okay, number 3, components are brought together to make new virus particles. So it's like an assembly of the units. The viral nucleic acid, the enzymes, and proteins are brought together and assembled into a new virus particles. Okay, so it creates and puts together a new virus. And then number 4 is your new virus particles are released and host cell then dies. Okay, so release of new virus particles, the newly formed virus particles are released from this infected cell, and that cell then, the host cell dies. This is actually called lysis. Okay. So what's happened here is the virus has duplicated itself, and then that cell dies, and then now you have these 2 new viruses going out and now going in and going into new cells, right? Now we've got 2 cells, and then they both duplicate, they die, and now you have 4 viruses going out. And so you see how it keeps spreading? Okay. All right, so those are the 4 steps. Whoa, sorry about that. Where did I go here? Here's the picture of it, okay? So there's 2 actual cycles here, okay, that we're going to look at. I was just seeing. Okay. So you have your lytic cycle and then your lysogenic cycle, okay? So before we go and I start and talking about this, so certain types of viruses, such as those that cause cancer, actually, are those that infect bacteria. They have a lysogenic cycle, which is this, okay, over here. Um, the virus does not kill the cell outright. Okay. It may coexist with the cell and be carried through many generations without apparent harm to the host. Okay. So it sits there. It's almost like it's dormant. Like it's just sitting in the cell, not doing anything. Okay. Um, and the virus, so like what we just went through, the viral replication, where it replicates itself. The virus enters the host cell, but instead of taking control, like I just explained in the other four steps, its nucleic acid actually becomes integrated into the bacterium's DNA. And you can see here on this side as we go through this. So it enters the cell and then it inserts itself into the bacterial chromosome. So it's inserting itself into that DNA. So when the cell goes to duplicate, it's now all of a sudden duplicated the DNA with the virus that's now instilled in it. Okay. Um, so it's now integrated into that DNA and acts as an additional set of genes, really. So it's now extra genes that have been put there. Then it's replicated along with the host DNA and it's passed along to the daughter cells. So it's replicated. Now you have two daughter cells with that virus now instilled in there. So during this period of normal replication with the cell, the virus appears to be in a dormant state. Okay. And then often the dormant virus may be activated by stimulus, such as an environmental change to a bacterium. So either temperature will affect it or pH. We talked about pH, that's the acidic and basic levels, or some other events such as changes in available nutrients. And then what happens is it's in these cells, but then these changes in temperature or pH are going to trigger a lytic cycle. And then once again, new virus particles are formed and released. Okay. So this is the cycle where it's dormant. And then all of a sudden something stimulates it or changes the pH or temperature or stress, depending on what the virus is. And it's now that's like it's activating those viruses. And now the viruses are going to start attacking the host cells. And actually, you know what another example of this is? Cold sores. People that have cold sores, that's a virus, right? And and people can I know my brother-in-law gets cold sores, and I know that he could go a year without a cold sore. And then all of a sudden, a stressful situation, you know. And there's a girl that I went to university with, and she used to get cold sores, and she would get them during exam time. And it was like clockwork. Every time we'd be starting prep for exams, she would get them because it's that stressful situation that takes the cold sore out of this cell cycle, the lysogenic cycle where it's dormant, and into this one where it's your lytic cycle. So that stressful environment switches it. So then all of a sudden that cold sore virus is on. Okay. So that's another example of the difference in cycles. You have this lysogenic cycle where it's dormant and hidden, but then something triggers it and it all of a sudden goes off. Okay. All right. So I think you're going to do, I know you hate these, but only one more gizmo on that one today. So something to think about. Some viruses, cancer, do not kill the host cell, but coexist with the cell and may be carried through many generations without harm to the host, which is your lysogenic cycle. All right. So treatment. So this is your viral infections. Okay. So most viral infections are difficult to treat. You cannot use antibiotics for viruses. Okay. Antibiotics are not used for viruses. So because some viruses are dormant for years before symptoms appear, so you don't even realize that they're there. Okay. So just thinking about so certain viruses okay, sorry, lost my train of thought here. So some viruses, so they remain dormant for years before disease symptoms appear. Certain viruses cause cancer because they add specific genes to the infected cell, which we just went over. And then therefore they turn it into a cancer cell, right? They've injected their genes. Vaccines. Sorry, I was trying to figure out the word. I could not, and I can't believe I forgot the word vaccine because I'm getting mine tomorrow. Vaccines are liquid preparation of the dead or weakened viral or bacterial cells that stimulate the body's immune system to fight back. So, some viral and bacterial diseases can be prevented with vaccines. So, a lot of the ones in the past before the coronavirus hit are, polio, smallpox, hepatitis B. These are all ones that are given orally or by injection. Okay. And these are a vaccine, just going over it again. A vaccine is a suspension. Remember suspension? You learn that in grade 9 when you're talking about like your mixtures, your homogeneous, heterogeneous mixtures. Suspension was one of them. So suspension, and it's prepared from dead or weakened viral or bacterial cells. Okay, so it's actually the actual virus weakened and it's injected into you to get your body to be able to identify and prepare so that if you actually get it, you can fight it off. I should just come forward. Vaccines are liquid preps of dead or weakened viral cells that stimulate the body's immune system to fight back. Okay, so vaccines can be injected or taken orally. And homework, you're not doing these questions. Okay, so that is it for I don't know why I keep losing my train of thought. For this, you're gonna do the virus quiz, and then you're going to do the virus gizmo, and then that's your homework for today. Okay. And that is it for today.